Okay, now we're going to move on to the topic of domestication and how humans are actually able to domesticate a lot of crops and animals. So the domestication of animals was possible because genes that control things like behavior we were able to choose and control through selective breeding. The domestication of bread wheat, Triticum aestivium, was similar. We selected two single genes, one that controlled the rachis, so the wheat would have a non-brittle rachis, so it would be easier to collect and wouldn't fall apart, so that we could harvest it and mill it into a whatever kind of milled down wheat is, and we wanted wheat with naked grains that lacked the fibrous hull that the wild type wheat has, that's supposed to protect the seeds. We don't want that fibrous hull because it makes it harder to access the seeds inside that we actually churn to produce the wheat product. So that's for wheat, controlling two genes to get our desired phenotype. Animals tend to be harder to domesticate, and we have only ever domesticated 14 species. And this is because the domestication of animals requires certain criteria. For example, the diet of the animals should be easily providable by humans. The animals should have short life cycles with fast growth times. They should be pleasantly natured. They should breed freely in captivity, so it's easy to produce more of them. And they should be docile with a follow the leader dominance hierarchy, so they would obey us. And they should also be comfort loving and not skittish in enclosures. And most importantly, these traits must be controlled by single genes or few genes that can be passed on to offspring, because it's pointless in us selecting these traits that we need for a good domestic animal if they're going to be majorly polygenic and we can't really control the phenotypes of the offspring through selective breeding. So if it's controlled by few genes, we take for example, from wolves, we take the wild wolves that we've got, we find the nice wolves in the bunch, we breed the nice wolves together, and we get nicer wolves, and then we breed the nicer wolves together, we get nicer and nicer wolves until we've got dogs. And then these traits are kind of polygenic. If you domesticate dogs, or if you domesticate foxes, although they're not fully domesticated, the domestication in the sense of giving them a good nature that loves humans and loves captivity, these kind of natured traits tend to come with other associated features. So they tend to have floppy ears and waggly tails, as well as being pretty good-natured and loving humans. So that's kind of something interesting to consider. So for these are more simple traits that are fairly easy to control, and we're able to effectively breed the kind of animals that we want to breed. But for complex traits, it's a bit more complicated. So Mendel's laws were figured out using discrete phenotypes controlled by a single locus, with either a dominant or a recessive allele in pea plants. These are pretty simple traits. Complex traits are when individuals cannot be put into discrete categories, as the traits tend to be quantitative or continuous. for example, height, and controlled by many genes, and we call this polygenic because many genes are responsible for the phenotype. Whereas for the more simpler traits, relatively few genes are responsible for the phenotype. So it's easier to domesticate, it's easier to produce offspring with the traits we want if those traits are controlled by few genes compared to controlled by many genes. Complex traits can also be altered via their environments, causing epigenetic changes to their phenotype. Human genetic disorders can be controlled by single genes, for example Huntington's, cystic fibrosis, Duchenne's muscular dystrophy, and sickle cell, or they can also be controlled by many genes, for example cardiac disease, Crohn's disease, cancer. Ordinarily, we are unable to discern the genotype from an organism just by observing its phenotype. We typically use a pedigree to work out the genotypes of a group of organisms over many generations. Most genetic discoveries consequently center around mutations that produce easily distinguishable mutants, for example Morgan's white-eyed Drosophila that we've talked about in the past. This is easy when you are dealing with phenotypes controlled by a single locus or few loci. For quantitative geneticists, however, researching complex traits, which remember are polygenic, controlled by many genes, phenotypes tend to be recorded over an entire population. For polygenic phenotypes, we have a few assumptions as well. We assume that each gene contributes the same amount to the phenotype. We assume that alleles are codominant, and we assume that two alleles can have opposing effects. So for the process of genotyping polygenic traits, the genetic components responsible for quantitative traits can be dissected via experiments that control environmental variation. They also randomize replicates and produce large numbers of progeny for their studies. A study of grain color in wheat showed a red to white color ratio of 63:1, with several distinct intermediate colors. The ratio suggests the trait is regulated by 3 genes, with 6 total alleles, because obviously you have 2 alleles for each gene on either homologous chromosome. And the phenotype suggests that they are all codominant. We produce phenotypic ratios that allow us to conclude how many genes are involved in the quantitative trait. We must first cross 2 pure lines that are homozygous for all 3 genes that we produce through a pedigree. So we use a pedigree to figure out 2 purebred homozygotes for homozygote dominant and a homozygote recessive. We breed these together to get an F1 cross, where all offspring are going to be heterozygous for all 3 genes. Then we breed these together to get the ratio that's going to tell us how many genes are responsible for the color of either being red or white, or if it's a mixture, some shade of pink. And the ratio is going to tell us how many genes are responsible. So if when we do our F2 cross, like the one that we see here, we get a ratio of 1 homozygote dominant, 1 homozygote recessive, and 62 heterozygotes in some form. And we don't really care about most of this, but when we're determining how many genes are responsible for the trait, we look at the ratio of homozygote dominant to the rest of it, and here we have a 1 in 64 homozygote dominant. And if we look at this, which we're going to go more into in detail next video, the 1 in 64 ratio from the F2, if we correlate it here, that implies that it's controlled by 3 genes.